Greetings to all of you and our respectable Madam Mazatul Ikma. I'm Norhidayat and these are my friends Noor Hafiza, Noor Artika and Noor Athirah Nabilah. So our topic is the production of steroids. Without further ado, let's get into the content.
The steroid compound, androstenedione and androstenedione, are direct metabolic precursors of testosterone, which are of special interest of industry. Different substrates can be used to produce ADD, which is androstenedione, such as cholesterol from wool, grease, or animal fat, sapogenins like hecogenins or diosgenins, and phytosterols such as beta-sitosterols or campestrols. Due to low cost and adequate conversion rates, phytosterols are appropriate substrates. They commonly appear in plants with high fat content, such as vegetable corn oils, soybean or rapeseed oils, tall oils, and waste materials of sugarcane or pepper pulp industry. Different production approaches for the bioconversion of phytosterols to steroid precursors, ADD, are available, like chemicals, semi-synthetic, or fully biochemical processes. For this case study, the biochemical conversion of phytosterols via the fermentation of the bacteria Fusarium species is investigated. Thus, different strategies are needed to increase the substrate solubility and with its microorganism accessibility. A promising approach is like using twin and lecithin cyclodextrins, or the addition of second phase such as propylene glycol, silicone oil, or vegetable oil such as soybean or sunflower oil. The application of second hydrophobic liquid phase to the fermentation medium is advantageous as the substrate concentration in the aqueous fermentation phase is kept constant at high level due to partition coefficient of the substrate between the phases. Furthermore, the product might be in situ extracted from the aqueous phase according to its partition coefficient, so that product inhibition by the microorganism is limited, as the upstream processing and the downstream processing should be investigated simultaneously. The consequences using different additives during fermentation need to be considered. To achieve an optimal solution for the whole process, the upstreaming process and the downstreaming process should be harmonized and not optimized. In the case study, the step was the decision of the additive during upstreaming processing. Different vegetable oils and cyclodextrins are investigated to improve the substrate solubility. Considering both the fermentation performance and the task for the downstreaming processing, vegetable oils are preferred to cyclodextrin, as they are cheaper, biodegradable, and contain naturally small amounts of phytosterols, and can be separated and recycled. Hence, for both the upstreaming and downstreaming processing, vegetable oils seems more promising, although the viscosity and flow behavior of the oil phase can be challenging. After screening several oils, soybean oil is evaluated as the best oil for a two-phase fermentation process to produce the target steroid precursor, AD.
Upstream processing is a part of a bioprocess that refers to the first step in which microbes or cells are grown, such as bacterial or mammalian cell lines, as in cell culture in bioreactors, for the fermentation for the valuable products to begin. Upstream processing comprises of inoculum preparation, media development, scaling up process, and inoculation. For the preparation of inoculum, screening is needed to find out which microorganisms are able to generate useful steroids. Once find out the microorganisms that undergoes selection process to determine on which one that yields more steroids product. Then, genetic modification takes place for the improvement of the microorganism's performance. As you can see from the table, Streptomyces albus yields 90 to 95% of steroid estrone, which is quite high. Next, the development of the media for the optimization of the growth parameters. Then, scale up of entire process where the culture volume is ramped up gradually. And lastly, the inoculum are inoculated into a bioreactor.
Alright, after scaling up process, you will take the culture into the bioreactor or fermenter. In bioreactor, the steroid will be harvested, which means the bacteria will grow in number and the product also will be generated. Okay, so what is bioreactor? Bioreactor is a device, or usually known as a vessel, which used to control or monitor the activity of biological catalysts to achieve a desired chemical transformations. Okay, here the transformation of steroid is conducted in stainless steel bioreactor of various sizes in a batch process. Continuous fermentation has not been used on industrial scale for bioconversion of steroid. This is because more attention has been paid to the continuous process in connection with immobilized cells. So here, we use aqueous media because the major limitations on steroid transformation are low water solubility, and it will cause specific difficulties in large-scale transformation in which the aim is high concentration of substrate. Therefore, the substrate is dissolved in properly chosen organic solvent like methanol, ethanol, or acetone, which it will precipitate to form fine crystals. So the size and shape of this crystal determine the rate of their dissolution in water. Also, the need to use immobilized cell or biocatalyst is because to minimize loss of enzyme activity and maintain longer half-life. Okay, so a suitable amount of steroid is 0.25 to 1 gram per liter will be added in a suitable solvent such as dimethylformamide, acetone or propylene glycol at the end of the growth phase. And also, the inhibitor will be added to avoid undesired enzyme activity. Culture growth and steroid transformation are carried out generally at the same temperature, usually between 28 to 32 degrees Celsius. Okay, so controlling the key parameter of a bioreactor such as temperature, pH, aeration, agitation are essential to maintain cells in a physical and chemical environment and optimizing their performance. So with the right condition of a bioreactor, the transformation of steroid usually will be formed between 1 or 5 days in the bioreactor. Steroids are analyzed in sample subtract from the tank in sterile conditions, and the process is regarded to be finished if the substrate is not found or if the residual substrate is on the same level in subsequent samples.
And I'm going to present the final part, which is the separation and recovery process of steroid. First is separation. So basically, separation is based on differences in the physical and chemical properties. So for example, polarity, boiling and melting temperature, size, and also charge. So the phases are separated, and the solid and oil phase are used as resieve after water removal. So the phases of the fermentation broth are treated individually as they contain different amount of target. For example, if the system consists of several phases and the product is not in each, the phases without target should be discarded or removed. So remove the largest quantitative contamination. So if one phase lacks target product, this phase is the main contamination. So furthermore, processing the more valuable phases reduce the overall volume of the material system and thus the scale of appliances and machines. As one separation step is mostly not sufficient to purify the product, additional steps or alternative is selected for the separation step. First, extraction to recover AD. So a HPLC method is developed to analyze all compounds necessary to calculate the yield and purity reached after a separation step. So the selection of adequate solvent is crucial for an efficient downstream processing and mass determination. So depending on the following steps like evaporation, solvents with a boiling point higher than the temperature limit to ensure a stable product are dismissed. This is because the target molecule will get damaged. So the listed solvent here can be appropriate to extract AD from the aqueous phase. However, for the oil phase, the binary solubility between the nonpolar solvent and the oil need to be considered. And it is also stated here that the best extraction performance is determined for using butyl acetate. Next is adsorption-desorption process to recover AD. So the target molecule located in fluid mixture selectively bind to a solid phase adsorbent and are selectively desorbed with a solvent. So suitable adsorbents are characterized by a large surface area and high capacity and selectivity towards the target compound. And adsorption-desorption process to purify AD is the most advantageous for the aqueous phase as the main amount of AD in the oil and the solid phase are not dissolved. So different adsorber particles according to their polarity are chosen as presented in the table here. Next, crystallization to recover AD. So crystallization is induced by supersaturation of the target molecule by cooling or evaporating the surrounding solution or adding an antisolvent. So after crystallization, the product is present in crystalline form of different habitus, needle and plates. Crystallization process are preferred where no additive are used as no subsequent solvent recovery and recycle are necessary. Next is purification by chromatography. So chromatography is another method used to purify AD. So the cost of this step is relatively higher compared to the value of the AD itself. So due to its low capacity, high operating cost, this process step should be applied late in the downstream processing. So the primary advantages of this technique is its ability to selectively separate compound from complex mixture. So purity of more than 99% for each molecule can be achieved.